Thank you very much, Dr. Bellinger, for very kind introduction. First of all, I'm very happy to be here and to talk to this Nobel Forum lecture series at the Karolinska Institute. So today, I'd like to talk about just overview of the proteasome. Today, my talk is 5 parts, but mainly I'd like to talk about in the second and third point. Introduction is just means to my research background. As you know, intercellular protein degradation can be divided two pathways. One is autophagy lysosome system. The other is Ubiquitin-Proteasome system. So I'm working for the proteasome for past 30 years. So therefore, today I talk about mainly in this system. If I have time to, I just summarize our recent work of autophagy studies. Just I told you how Ubiquitin-Proteasome system is discovered. As you know, a long time ago, M.V. Simpson found metabolic energy is required for protein degradation. But this is an unusual observation. So nobody believed this proposal. But 25 years later, Dr. Alfred Goldberg found cell-free system using of reticulocyte lysate. As you know, see on the right, just ATP stimulate greatly the degradation of abnormal protein. This is required ATP energy because non-hydrolyzable ATP does not affect this stimulation. But you know, next year, Dr. Abraham Haskell and Aaron Tsikorova's group found the Ubiquitin system. I don't tell you the details of that, but only one point is this is Ubiquitin, this very small protein is ligated by isopeptide bond, and then Ubiquitin to repeatedly covalently attachment of like this. And this reaction is required ATP hydrolysis. This is mimic to protein synthesis. So therefore, in this case, overall is the protein degradation, but the mechanism is very similar to protein biosynthesis. That means to easily exploration of energy dependence. And then 1981 to 1983, I in Tokushima is my native town, and I went to United States, Alfred Goldberg laboratory 2 years. During this time, I have found just summarized is this slide. As I told you, ATP is required for ubiquitination reaction. However, we found in this time degradation of ubiquitinated protein still requires totally ATP energy. So, and later this is characterized ATP-dependent protein complex. Later we named this is the proteasome. So in second part, I am focusing on structural analysis of proteasomes because usually proteolysis is a simple reaction catalyzed by trophic chemotrophin. In this case, the size of protein very, very small, 20-30 kilodalton. However, proteasome is very huge complex, 2.5 million dalton. That means so we named this somes is resembling of ribosome. So this means the proteasome is a very big protein. So, but this also at that time nobody believed the existence of that large protease. So we focused to about 15 years to analyze this complex. I took for the first time, 1991, this is an electron micrograph taken by after negative staining with uranyl acetate. As you can see, this is a structure of so-called 26S proteasome, active proteasome. In that time, I just drew in the cartoon that proteasome, this type of dumbbell-like structure. After then, we have been collaborated with over 20 years, Dr. Wolfgang Baumeister, Max Planck Institute. This is a current most cutting-edge structure of the 26 proteasome. This by analysis, single molecule analysis by cryo-EM and X-ray crystallography. So active proteasome can divide to central catalytic particle. This is also known as a 20S proteasome. And active proteasome consists of the binding regulatory particle, RP, in both sides. So this is a most modern structure of proteasome. So initially we purified proteasome and analyzed to the dimensional phase. I could see the over 30 spots. And then we have to isolate individual components. And then the primary structure have been determined by Edelman degradation and finally the shedding cloning. So in most subunit, almost 70% subunit, we have determined the primary structure by shedding cloning. Now this is a full subunit composition. As you can see, catalytic particle contains two alpha and beta subunit, like this structure. On the regulatory particle, it consists of three kinds of subunit. One is ATPase subunit. It's called RPT. RPT means to RP, triple ATPase subunit. This consists of 6 which to make that ring-like structure. Another is non-ATPase subunit, like this. So we have succeeded to crystallize a mammalian 20S proteasome. As you can see, the structure alpha different but related subunit to form the ring, alpha ring, beta ring, alpha ring. And this is a very unique protease. So far, the proteases are divided to the 4 processes, serine, cysteine, aspartic metal protease. But proteasome is first identified as any terminal threonine protease. It's based in novel protease. And also, 3 active sites are located in the cavity of the beta ring. 3 active sites means that is beta 1, beta 2, beta 5, which are specificity of peptide cleavage, and so called to caspase-like, trypsin-like, chymotrypsin-like activity. This is the degradation of in the acidic and basic and hydrophobic amino acid residues. And that is regulatory particle. So far, we have challenging the crystallization, but all we fail to crystal. Not only us, but also other groups, because this is very fragile structure. So therefore, now we are collaborating with Dr. Baumeister and analyzed by cryo-EM. So as you can see, these two RP complex contain two unique subunit. One is the ubiquitin receptor, RPN10, RPN13, which can able to trap polyubiquitinated protein. And also ubiquitin is very important. So for recycling of them, three the ubiquitinase are associated in this complex. The ubiquitin is cleaved after trapping to and for recycling. But the protein are degraded by in the central part. But usually cytosolic protein have tertiary structure. But this tunnel of alpha ring is very narrow. So therefore, trap substrate should be denatured. In that case, ATPase function to destroy the tertiary structure. This is reason for energy requirement. So this is the overall structure. In this structure, we have obtained by cryo-EM. But quite recently, we have collaborated with Toshio Ando, Kansas University. He devised high-speed atomic force microscope. This is one molecule of 26 proteasome. This AFM also can detect to this tetraubiquitin. So therefore, we are now in vitro in the visually how 26 proteasome degrade the polyubiquitin protein. So we are now trying to that's the mechanism of the proteolysis. We are also interested to analyze assembling the mechanism. It is a long-standing question how the complex structures of the 26 proteasome are organized in a high fidelity. So 10 years ago, we have started to this project. I have no time here to discuss the details. Just summarize. This assembly pathway of 20th proteasome. We found proteasome dedicated chaperone, which assists proteasome assembly. 2005, we found PAC means proteasome assembly chaperone. 1, 2 heterodimer and 3, 4 heterodimer. These two dimers chaperone are required for alpha ring formation. And for assembly of the beta ring, it's quite a different way to each subunit to step-by-step attachment on alpha ring. In this case, N-terminal propeptides function as intramolecular chaperone to assist beta ring formation. How about the regulatory particle? This is just to summarize the assembly pathway of the RP complex. Fortunately, we found 4 RP-specific chaperone. We named the DAC 1, 2, 3, 4. And also, we found UBP6 or other bug 6, other chaperone-like function are involved in the process. Quite recently, other group, found another chaperone, ADC17, which is required for this subcomponent of this complex. And we have a group of structural biologists. We have determined almost all chaperone structure like this. And also, we have, for example, PAC3, PAC4 co-crystallized with interacting protein alpha 5 subunit. As you can see here, this is superimposed in the 20th proteasome. So therefore, chaperone is associated like this, and then promote to the assembly alpha ring like this. So this overall structure. So what we have found 10 proteasome associated chaperone. So we think this is a very new concept of macromolecular assembly. As you know, in cell has many complex, typically ribosome, spliceosome, many complex, but nobody knows assembly mechanisms. We succeeded to propose the new concept, so therefore our result may influence to other studies to elucidate the assembling mechanisms. So I'd like to talk about the third part in the physiopathology. We have been studying in cell cycle or stress response of proteases, but today only I focusing on immunity. This is my main subject. So as you know, in cell-mediated immunity, so this is not antibody formation, but for example, to viral protein or tumor antigen. So in 1990, before then, this non-cell protein associated class 1 molecule to present to killer T cell to kill. So therefore, this is very important to discriminate cell or non-cell. However, mechanism to presentation is unknown. However, 1990, top transporter was found of 4 groups, United States and European immunologists. They found cytosolic peptide transferred by this peptide bond and a peptide associated class of molecule. So therefore, people, including mainly immunologists, believe the antigenic peptides are produced by cytosol. In 2 years later, 2 or 3 years later, Alfred Goldberg-Kenlock group using the proteasome inhibitor, and they found the proteasome is a processing enzyme. On the other hand, as you can see here, this is a TCR recognized non-self peptide and also together with MHC molecule. So therefore, that antigenic peptide or CTL epitope are very, very important discrimination of self and non-self. We fortunately found Immunoproteasome. I'd like to talk about shortly in this strategy. In the cell treatment with gamma interferon usually say to the immunocytokine. And then after treatment, the gamma interferon for a couple hours. And then cell protein are leveled by methionine, and then purified by using antibody. We surprisingly found, in that time we named XYZ. These 3 spots disappeared after treatment interferon gamma. On the other hand, 3 new spots were appeared, LP7, LP2, MECL1, now to be renamed. So we have cloned all 6 spots by ctDNA cloning and found these are catalytic subunit. And beta-1, beta-2, beta-5 is very homologous. Proteasome itself are very similar, but homology is almost 10 or 20%. However, in this case, similarity, identity over the 60%. So therefore, we propose gamma interferon stimulates cell in the constitutive proteasome to exchange by interferon gamma inducible proteasome. And then this is inducible and also related to immune response. So we named this the Immunoproteasomes. And as you can see, this is a peptide to associated with the model in immunology. Very important point of C-terminal is the anchor of the MH class 1. And also middle part is important. As you can see, proteasome change to immunoproteasome is catalytically high chymotrin-like activity and low caspase-like activity. As you can see, the pocket in C-terminal amino acid residue is almost all hydrophobic amino acid. So that means the immunoproteasome is very good to produce to MH1 class 1 associated peptide. This is a cartoon is antigen presentation for T-cell response. During our recently, we also found another type of immunoproteasome. We named that in the thymo-proteasome. In the, searching in the EST, this is cDNA information. In 2007, in that time, a student, Shigeo Murata, found some unrecognized new subunit was expressed largely only the thymus. For example, this protein is just a gene located in beta 5 chymotrin-like gene in very closely that opposite orientation. But this gene is intron-less, so that encoded by a single exon. And then we raise the antibody. As you can see here clearly, this new subunit is only expressed in the thymus. So therefore, we named this by beta 5 Ts. And then we have identified the partner active site subunit. I show in the, this beta 5T contained proteases also contained beta 5I beta 2I, the immunoproteasome subunit. So therefore, we referred this complex is proteasome. What is thymus? Thymus is essential for development of T lymphocyte. In cellular level, this is discriminated, self and non-self. More deeply, I explained that immature thymocyte, the double negative cell, in the no expression of CD8, CD4 co-receptor, and these cells develop in the cortex. Thymus is inside medulla, outside cortex. And then during development of double positive cell, in this process, TCR rearrangements occurred. So therefore, large number of double positive cells are formed. And then double positive cell to in the developed single positive cell. In this case, usually people say that this is an education. That means a two process. One is positive selection, another is negative selection. So we test in vivo function of the proteasome. So we make beta 5T knockout mouse and the venous knockout mouse. As you can see here, in the thymo-proteasome is only expressed in thymus and only expressed in the cortex. How about the phenotype? As you can see, we couldn't see any difference of the architecture of thymus and the size. But only difference, the deletion of beta 5T, in this case, instead of beta 5T in the beta 5I appears like this. So therefore, biochemically only one data we obtain that is conversion of thymo-proteasome to immunoproteasome. Most important point is the thymocyte, analyzed in the, by factor analysis. As you can see, CD double negative, double positive, double CD4 single cell, their amount remained unchanged. On the other hand, as you can see here, CDA T lymphocyte reduced greatly. So that means to thymo-proteasome really involved in positive selection. And we have tested many things, but we couldn't see any, we didn't find any difference except in the two phenomena. Two phenomena mean to the conversion of immunoproteasome and also decrease of killer T cell. So what is the pathological function? We have tested with influenza virus infection. As you know, wild type cell is a key to cell-mediated immunity system. So we couldn't show any death of mice. However, beta 5T lacking mice, as you can see here, mice gradually death. So that means to this beta 5T is very important in the maturation of T cell development, T cell repertoire formation. And so therefore, the cartoon in our proposal. So in the cortex, particularly in the CTEC, is the thymo-proteasome produce peptide which are assisted positive selection like this. On the other hand, beta 5T deficient CTEC, in this case, the immunoproteasome rich in CTEC. This immunoproteasome also produce a peptide, but this peptide cannot promote positive selection. So therefore, thymo-proteasome is very important, involved in the T cell development. That means to cell-mediated immunity. It's the genetics is okay, but we want to know the mechanisms. How do thymo-proteasomes work catalytically in the thymus? This work done by collaborators Murata and Takamata Tokushima University. This is an unpublished result. As you know, a mechanism of positive negative selection. A long time ago, immunologists proposed to avidity or affinity model. So double positive cell is in the exposed T cell receptor outside, and the interaction with the CTEC. If double positive cell has non-functional TCR, in this case, over the 90% double positive cell death by apoptosis by so-called null selection. And if CTEC to express own antigen, in that case, this interaction is very tight. So in this case occur by negative selection. So what is the positive selection? People say this is intermediate, moderate ability. So interaction is not too high, not too low interaction. So people reviewed many of this model, but nobody provide evidence. Our thymo-proteasome may produce peptide fit of this mechanism. We test this in cell. We have made it only expressed to immunoproteasome and thymo-proteasome, and then extract by analyzed glacial density gradient centrifugation. It's a usual way to assay of proteasome. As you can see here, trypsin-like hypoproteasome activity remained unchanged. However, beta-5T, the thymo-proteasome containing cell, this activity is greatly reduced. So therefore, thymo-proteasome function, the primary structure is very similar to beta-5 or beta-5I, but function is quite different. So we have tested that in the cell we made to beta-5I cell, beta-5T only expression cell. And then we have to immunopurify of two major mouse plasma molecules. And then their peptides were analyzed by mass analysis. This is just cartoon. This is a position, the hydrolyzed product of immunoproteasome in the thymo-proteasome. But we found curiously in thymo-proteasome produced to intermediate region in the high frequencies of protein residue and also acidic residue. So like this is just cartoon. Again, I told you in the antigen peptide is very important to in the TCR recognized. In that case, these antigen peptide properties are quite different. So to demonstrate this is really involved in the positive selection. In this Takahama's work, this two T-cell immunologist. RTOC assay, this is an in vitro developmental assay of the organ culture. For example, in this case, overwhelming antigenic peptide is very famous peptide. And immunologists know non-positive selector. Positive selector means, in this case, force change the glycine residue. That explains this. This is a no peptide. If addition of overwhelming peptide does not change to a killer T-cell level, however, positive selector is changed to Z4. As you can see, increase the twofold amount of CD8 T-cell. But surprisingly, change to protein here, 40% development. This is surprisingly large amount of stimulation of organ culture level. So that means to also to other acidic amylase residue also promote, but not greatly. So therefore, we think about it's a cartoon, right? Immunoproteasome and thymo-proteasome produce different kind of peptide. In particularly, thymo-proteasome produce, and to that peptide contain protein residue. Protein residue maybe to this interaction not strong. So there was a reason we believe this why thymo-proteasome are responsible cell-mediated immunity during T-cell position. This is a famous T-cell researcher in Ukraine, in Germany, and Hoxis in American, United States, they just reviewed 2 months ago, and they to address the importance thymo-proteasome positive selection. A similar case is class 2 case is similar story, but in this case, different catepsin are involved in the production of class 1 to peptide for CTEC only. So therefore, this is general aspect of immunity in the not only to CD8 T cell production, but also CD4 point T cell production. So now, just summary, we found the 1988 proteasome, 1994 we found proteasome, immunoproteasome, and now we found the thymo-proteasome. These proteasomes are very important because of this is the evolutionary tree. As you can see, these genes are produced similarly to many of adaptive immunology genes. So therefore, I guess thymo-proteasome or immunoproteasome are important in the immune responses. Finally, I talk about selective autophagy. As you know, autophagy is now fashionable field in life science. But as you know, autophagy means to in the some small particle, autophagosome appears suddenly in response of starvation. And then, you know, the lysosome fused with autophagosome and then degrade into trapped to materials, including protein and organelles. This observation is 1962. In this case, rat fasting one day, as you can see here, autophagosome, just lysosome is fusion. This is like in cell culture level, just 4 hours starvation, or in this hepatocyte cell appeared large amount of autophagosome. So therefore, autophagy is necessary for starvation response to obtain the nutrient for amino acid to make macromolecule or energy supply. However, we have is commerce group to found proteasome autophagy is highly related. Recently, we made to liver-specific proteasome-deficient mice. As you can see, in this case, protein aggregate formation like this. And this is perhaps non-functional mitochondria. If we do not manage the autophagy itself, but decrease of proteasome function, as you can see, autophagy appeared like this. So therefore, two product systems may be interrelated. So we genetic analysis by using ATG7 is essential for autophagy. This is rat liver. In this case is nutrient rich. However, as you can see, liver size increase largely. So loss of autophagy leads to hematomegaly, and also over 1 year later, 100% efficiency to generate tumor, liver tumor like this. I have no time to discuss the mechanisms, but we know that the two pathways operated in tumorigenesis. One is the loss of mitochondria, functional analysis of mitochondria decreased greatly after autophagy deficiency. And also, NRF antioxidant response system to increase greatly. So this is one of the reason of phenotype, even in rich condition. And also, we have analyzed to make autophagy deficiency in central nervous system. In that case, as you can see, in the neurodegeneration occurred in like this. This is a big surprise. So far in the Alzheimer's disease or Parkinson's disease, in that case, some toxic protein are accumulated. However, in this condition, just decreased autophagy is caused to neurodegenerative disease. And then analyzed in the brain, as you can see, in the neuron contained large amount of aggregate. And surprisingly, these aggregates are stained strongly by ubiquitin. It is a little bit of shock for me because so far ubiquitination is a degradation signal for proteasome. However, ubiquitination is also involved in selective degradation by autophagy. So therefore, both systems are interrelation. The key molecule is ubiquitin. And also, we have examined Parkinson's disease. This Parkin is because we have found 2000 Parkin is ubiquitin ligase, and the PINK1 is a protein kinase. But 2004, and this is more, I guess, more than 20 years ago, this gene are co-subgene of Parkinson's disease. This is autosomal recessive. That means the loss of function of two protein causes to early onset Parkinson's disease. Just I summarize its function. We found to PINK1 and Parkin. PINK1 is usually in degrade, but therefore we couldn't detect it. Normal mitochondria. Normal means to in the high energy production in the mitochondria. However, in the depolarized mitochondria treatment with CCCP and capillary, in that case, degradation was inhibited. And also, we have found this PINK1 is complex to Thomson translocator and doubly autophosphorated. Therefore, this active PINK1 promotes Parkin, cytosolic Parkin to impaired mitochondria. In this case, cytosolic Parkin is latent, but recruited Parkin is activated. So we have examined the mechanisms of activation with this. This data is very clear. PINK1 is located upstream to same signaling pathway of downstream element of the Parkin. And quite recently, just 2 months ago, we succeeded to identify the mechanisms of activation. So initially we found, also other group found to Parkin phosphorylated N-terminal ubiquitin-like domain. But in this case, only activation partially. We have another target, Another target is surprisingly the ubiquitin. Ubiquitin also phosphorylated by PINK1. In that case, Parkin activated freely. So this is overall busy slide, but just we found that regulatory mechanisms of PINK1 are also regulatory mechanisms of Parkin. So far, many mutations have been reported, but the hotspot is only these processes. So therefore, its regulation of two protein kinase ubiquitin ligase is very important. Quite recently, Tony Hunter reviewed our study by PINK1 the first ubiquitin kinase. So that means to activate PINK1, again to phosphorylate two proteins activate, and then activate Parkin to ubiquitinate outer membrane of mitochondria. In that case, autophagy attack this damaged protein to degrade. As you know, mitochondria is autonomously growing. So therefore, impaired autophagy was destroyed. That's enough to keep the healthy in mitochondria. This is a key point of impairment of this process cause of neuronal death and finally Parkinson's disease. So therefore, autophagy, long time to people say the importance of adaptive immunity to starvation induction. But quite recently, constitutive autophagy, it means selective via the ubiquitination. That's important to like this. We have published many papers on this. And then perspectives, well, I have no time, so there are only one slide, not just this publication of Ubiquitin-Proteasome system, this autophagy system. As you can see, ubiquitin was identified in 1978 something. Proteasome discovered 1988, and we discovered Immunoproteasome 1991, the Thymo-proteasome 2007. And also we have examined the contribution to structural analysis. Almost all primary structure of Subunit, and recently in the tertiary structure, the collaboration with biomedical group. And autophagy is also to, autophagy gene identified in the 1990s. And then papers increased dramatically. So now the both systems are very important in life sciences in general. So this is our collaborator, our group. We have collaborated many groups. So they were just I thank for them. Finally, our institute is Tokyo. Tokyo, the Shinjuku is here, very close to 50 minutes from Shinjuku. So therefore, you have a chance to come to Tokyo, drop up our institute. Thank you very much.